and welcome to your virtual tour highlighting restoration and resource management initiatives in the Commonwealth of the Northern Mariana Islands. The Commonwealth of the Northern Mariana Islands, or CNMI, is a tropical archipelago in the western North Pacific on the border of the Philippine Sea and the deepest place on Earth, the Marianas Trench. The vast majority of the population resides on the islands of Saipan, Tinian, and Rota. However, the islands extend about 400 nautical miles from Rota in the south to the most northern island of Uraques or Farallon de Pajaros. The islands and coastal resources of the CNMI are incredibly diverse, as is the history and the culture of our region. In fact, the CNMI is the only United States Commonwealth with 3 official state languages. Today, on this virtual field trip, compiled for the Society of Ecological Restoration's 2021 World Conference on Ecological Restoration, subject matter experts from resource management agencies, nonprofits, and educational institutions will be sharing current project highlights from our region. Contact information will be posted during the questions and answer sessions at the end of this video tour. Today, we will be highlighting watershed management planning efforts on the inhabited islands of Saipan and Rota, as well as species and habitat conservation and restoration efforts throughout the chain with a focus on endangered and endemic species recovery on Rota and in the Northern Islands. Together, these efforts are supporting cross-cutting management goals to support sustainable stewardship and use of our marine and terrestrial resources. We will start your tour on Saipan, the largest and most populated island in the CNMI, stretching about 12 miles long and covering about 45 square miles. Residents and tourists alike are drawn to Saipan in part because of the island's beautiful beaches and striking shoreline attractions. Supported by the Federal Coastal Resource Management Program, Ridge to Reef management planning efforts have been ongoing with a focus on high-value and high-use resources since 1983, when the CNMI adopted the Coastal Resource Management Act, creating the Coastal Resource Management Office. Today, you will be hearing from resource management specialists about ongoing Ridge to Reef conservation efforts underway throughout the CNMI with a focus on priority watersheds. Conservation and restoration experts from the Department of Lands and Natural Resources, as well as nonprofit and educational organizations on their current goals and implementation efforts. Together, these conservation actions reflect overarching sustainable development goals that the CNMI is working to further. These include taking urgent climate actions and promoting data collection and analysis to support sustainable use and conservation of our marine and terrestrial resources. There are so many partners doing amazing work to promote these and other sustainable growth goals, many of which connect to larger regional and international management efforts. We hope today's virtual field trip will help connect subject matter experts and engage conservation professionals to help amplify our local efforts and build networks to support this work. Thank you, Society for Ecological Restoration, and you, our field trip attendees, for this opportunity to virtually show you the places and resources we are working to protect here in the CNMI.
DCRM's Coral Reef Initiative, we not only conserve and restore coral reefs directly, but take into consideration the wider terrestrial landscape and its influences on nearshore ecosystems. At the center of this ridge to reef approach are our island's watersheds. Saipan, for example, has 11 delineated watersheds. Among these, we have selected 4 as priority watersheds for our land-based coral conservation efforts. These were selected based off of their biological, economic, social, and cultural significance, and are the West Tapachao watershed, including the Garapan subwatershed, the Lau Lau Bay watershed, as well as the Achugao watershed right here, all on Saipan, and in addition, the Talakhaya watershed on Rota. We take a collaborative approach to watersheds management and invite participation from various partners, including local and federal government agencies, NGOs, nonprofits, academia, and community groups. As watershed coordinator, my role is to coordinate these groups and their efforts to best tackle watershed issues. This is perhaps best encapsulated in our watershed working group, which meets regularly to align natural resource management, conservation, and development initiatives, to provide technical guidance and project support, and to share information and research in the CNMI. This is a holistic approach to watersheds management in terms of land and marine-based natural resource management, but also holistic in a sense that it invites participation from a diverse group of resource users, managers, researchers, etc. It is important in planning to consider environmental threats and issues we have on an island and incorporate solutions, such as wetlands restoration to reduce urban flooding. Another threat we have on island is that of fire and issues with habitat loss, sedimentation, and damage to human infrastructure.
Fire poses one of the largest barriers to national resource management on Saipan. It is estimated that between the years of 2013 and 2020, that over 2,900 acres of land have burned in Saipan. And this ranges from the very northern point of the island near Cowtown and Marpi to the very southern points of the island around Nafton and Coral Ocean Point. Where we are standing right here at Wireless Ridge, this grassland has burnt on 3 separate occasions within this time frame. Grasslands are one of the primary vulnerable land cover types to fire, but many other land cover types also burn with increasing frequency. These land cover types include scrub shrub, pasture, and some forest areas. When fire runs through a grassland such as this one, it leaves bare exposed soil that during the next precipitation event can run off into streams and creeks, which eventually lead into the ocean. Once the sediment and soil ends up in the ocean, it can cover coral reefs. When the soil and other pollutants cover coral reefs, they are less likely to reproduce, less likely to feed, and less likely to photosynthesize or have less ability to photosynthesize. And because of that, we feel that fire and land-based sources of pollution are one of the primary drivers to coral degradation in Saipan and within the CNMI. Outside of coral degradation, fires also represent one of the primary drivers for invasive grassland species spread. When a fire occurs in a grassland such as this, the dry grass carries flames until it reaches the forest boundary that you see right here. However, each time a fire occurs, the fire penetrates into the forest cover just a little bit further. This is because when the fire hits the forest, a little part of the boundary gets burned. And because grassland species recover at a rate that is faster than the native tree species, that little sliver of forest boundary turns into grassland species. And as such, what we get is a positive feedback loop where fires change forest cover into grasslands, thus allowing for more fires to start in the future. And so you have an ever-expanding grassland and an ever-retreating forest boundary. And this results in further degradation of the forest, further expansion of the grasslands, and eventually more sedimentation of the coral reefs. The vast majority of fires on Saipan occur during the dry season, when precipitation is at an annual low. During this time, vegetation such as grass, trees, tongan, are all very vulnerable to fire. On Saipan, it is estimated or predicted that most of the fires are caused by things such as lit cigarettes, barbecues, and brush burning. However, there are a couple of other theories that include lightning strikes, hot cars parked on dry grass, and broken glass that is located in the vegetation might also cause fires in the grasslands. Current research is looking at the spatial and temporal patterns of fire on Saipan and the greater CNMI. What we have found is that there are 4 hotspots in Saipan that seem to represent where the vast majority of fires occur. And it's no surprise that these 4 hotspots are where the largest grasslands and the most concentrated grasslands are. What the research has also shown are that grasslands in urban areas burn more frequently than rural areas. And this is because, or this is thought to be due to the greater number of people which means there are more cigarettes, more barbecues, and more brush burning that would result in more number of fires.
The Achugao Watershed is our most recently selected priority watershed, with issues of land-based source of pollution, and particularly erosion and siltation, being of chief concern. Although development of the watershed management plan is still underway, we're already moving forward with implementing projects meant to address issues of upland erosion. Last year, DCRM was awarded a grant from the Department of the Interior to reforest parts of a 140-acre grassland on the western slopes of Saipan within the Achugao watershed. This grant will fund 10,800 trees, or at least 6 acres of reforestation, converting open grasslands into native limestone forest. To accomplish this, we are working with local partners, including the Department of Lands and Natural Resources Division of Forestry, who are leading plant propagation and providing technical assistance, and the Mariana Islands Nature Alliance, or MINA, a local environmental nonprofit, who are assisting DCRM with site preparation and maintenance, outplanting, and monitoring. Similar projects have been carried out here in the past, on Saipan in the Lau Lau Bay watershed, and on Rota in the Talakhaya watershed, still underway today. Both have seen great success. The Lau Lau site has many well-established trees, and we're beginning to witness natural succession among the planted trees, such as this Philippines ground orchid. We are using those experiences and lessons to inform our planning with this project. And at Achugao, we are lucky not to have badlands, or areas of exposed soil, that must first be remediated, so we plan to jump forward with native tree planting. At present, final siting is underway. We intend to plant within a larger 140-acre public land parcel in the uplands of Achugao, here in what is locally known as Wireless Ridge. As Elon highlighted earlier, open grassland is prone to fire, and we anticipate a reduction in fire vulnerability once trees become established and mature. While upland restoration is only just getting started here in the Achugao watershed, on Rota there's a reforestation project that's been going on for over a decade. The work being done in the Talakhaya watershed is very much an inspiration for what is possible with environmental restoration and conservation here in the CNMI.
Our virtual tour now takes you to Rota, or Luta in Chamorro, also known as the Friendly Island. It is the southernmost island of the CNMI and the second southernmost of the Marianas archipelago. It is home to numerous endemic species. Conservation efforts are focusing on habitat restoration,
captive breeding for the aga,
rehabilitation for the fanihi, turtle monitoring, bird repatriation, invasive species control, and more.
Afaday, welcome to the island of Rota. My name is James C. Manglonia, and I work for the Department of Lands and Natural Resources under the Division of Agriculture Forestry program. I'm here today to talk about the Talakhaya Watershed Revegetation Project. The Talakhaya Watershed Revegetation Project basically started back in 2007 with funding assistance from NOAA Coral Reef Initiative Conservation Program. So for the past 13 years, NOAA was the main funder of this project. For the past 2 years, we have transitioned to the USDA Forest Service Forest Stewardship funding mechanism. So it's been 15 years in the making since we have implemented the Talakhaya Watershed Revegetation Project. So the main objective of the project basically is to try to revegetate those barren lands. Some people call it badlands, you know, there's so many names, but it is referred as barren lands. And like I said, our main objective was to, you know, reintroduce a vegetative cover so that we try to minimize, you know, soil movement from. This is a project, like I said, under CRI conservation program that deals both with terrestrial and marine. So the project has been in the making with assistance from the community, volunteers, participating in the project, outplanting basically grass and forestry species. So for the past 13 years, our main objective was to replenish those barren lands and try to bring back to its original state. And for the past 2 years, we've been transitioning into, outplanting native trees. So our objective basically is to transition and to convert those grasslands into forest lands. So we've been using, for the past 13 years, we've been using nitrogen fixing trees, specifically acacia confusa. So that's the species that we've been using for many years in combination with vetiver grass. So vetiver grass, basically we use that along the counter lines to hold, catch soil, trap sediment, and hopefully in time will create a soil buildup so that native grasses and other native trees will be able to regenerate on their own. So above those vetiver rows, that's where we basically plant those legumes, the acacia confusa seedlings. And since vetiver does not tolerate shade, eventually over time it will be shaded out by those, acacia trees. So basically that's our what we've been doing for the past 15 years. And this is an effort where the community has participated tremendously. In previous years, we have involved, the schools and community volunteers in the all, aspects of the project, from preparing the sites to transporting, planting materials to the actual outplanting of seedlings. So this is a project where it's pretty much well known, I would say, not locally, but perhaps regionally and perhaps internationally to some extent. So we are proud of what we have accomplished so far, and this is a project that basically has shown some success, and hopefully other regions will be able to use us as a model for others to follow in other areas where they have similar problems with similar problems in highly erodible lands. So that's what we have with us, so that we would like to share with you folks. So hopefully you gain some insights as to what we've been doing for many years. So I extend undang kulo na si Zeus Masi for the opportunity to give some insight. Thank you. Once again, from Rhoda, Afaaday, to all of you participants. Buenos dias, Afanielos dan Afanasay na, Gaigizo Pago, Palanga dan si Senior James Bamba, no Pago na ani, Gofaan, Unito Isamnag, Pago na ani, no Ami dan ni grupon mami, Ikadgon Pago na ani para bempan mananom, si Mizan Zoga, Istina Tronco, Natibo Paitano, Maulig na trongko para todo siya gaga, particularmente fanihi dan paharo siya gini gitanota, izaluta dan noti Marianas. Este na lugar intanunumi siya na natibo na tronco cosa kisinya ta ataha mas problema gini kita nota especialmente ibandan nilak siya naman kinikilili papa esta revegetation na programa no kahit tempo esta na masusedi sa komunidad luta manasaw no anay para maagwiguzi na hinano anay para siya tumanumi siya megay na klasin tronco para beneficyon gaga matsolik zanhita na taw tano gini kita nota islan luta na tinanom izoga may para ben tanom sempi undia fuera kipago mandongkulo sa produsi siya megay na beneficyo para gaga parewa para sagan no ningkano sombra zan fanlihengan posistige proposito nesti na no inano para ta continua to manumis siya ipalanga no diferentes klasen tronco na lugar indisigna si small gidania geftano ilugar pentanunumi siya na tibo na tronco no ala kwenta estados lutsa na no tinanom zoga sempi giman mamamayla siya na dia bay continua to manumi otro siya no na klasen tronco tad ko mo dogdog no lemay dan amot siya na ito siya na tronco mamproduce ng kano para ipaharo especialmente palumata siya gini no itoto kurbata sali dan no somnak pues sinimbad ko bay continua kita gini na manera pues sige debi dan antes para ta sogit si sota na debi ta pipare lugar dan ginunatan ni reason mami inli na inkela di lugar sis defecton problema asuntun binado gini na lugar necessario ko na binanombo ay para sige na debi pipare lugar Dorantin tempon maipi law sinya wa mananon gi masya hafa na tempo maipi tempon anglo pag tempon sa waha no sinyong sogi para na wa libiu de trungkomo ti mampusin afekta na iklema pues ano natan tinanom mami para bentanom ala kwenta 2 PA media no nilikanya pues na material no zanginon mentrason na mas hanom mas domangkulo na artikulo za manusteni mas hanom kosaki itinanom sinya kidurantin tempon maipi gumaha no alibyo para guiza para itinanom necesaro no hujung mampu si hali gagay gya hanom gumagwa nay dimagang moisture no okay masya ti no afazi ti mampus no mesgo ilugar launahong moisture na isinya itinanom no lala gidurantin tempon may pues estigi inisa siya na lugar mucho mas palangan mampus anglo dyan may pina lugar después 4 grano na bono is slow release adumidi di fertilizanya sempi umanaw ka to tronco tigopes pues na klase na bono sinya inasistik tinanom 3-4 meses na tempo actually half paron tanom na ay gwini na Gigi oda unos cuantos oda orizanya may tetinan na oda may gayi gitano ay gwena binan mama mi gwini no Talakhaya na banda pues istige no mensahe para taw-taw ta gini gitanota zan taw-taw san hizo ay istige binan mama mi gini inajujuda inagwahati para konserbasyon no todo riqueza Rikesan tano na para ufang gagay giya para man mamamayla siya na kinawulong. Istigi patigi obligasyon mami como taw tano para ba in pasa papa hafa tiningo mami da pwede iman mamamayla na pinipit siya pinipit mo na hafa pa fan matsogi necessary na susod pa fan matsogi ano para mag-continue pandan sa konserbasyon sin mas felicitamos si Jesus Masi por toda a atenção medida bienvenido adiós.
Buenas and Afade todos Samson ni Manmafatu para Society of Ecological Restoration 2021 na virtual field trip pago na dia. Ko hijos midzu si James C. Bamba sa Mangaigi pago gi mas gatpon na isla gunigi sa Marianas, Islan Luta. Man matututsuham gi Departamento Tono and Riqueza Natural, DLNR gi Bandan Forestry sa hami yung pati gi programan manhasan na species. Si Zusma si talo ni Fanaton Mizu pago sa nihi tafan hono tafan picnic. On behalf of myself and my two colleagues, we'd like to welcome you all again to our virtual field trip today to visit several of our work sites on the beautiful island of Luta. Myself, James Bamba, the nursery technician, along with my two colleagues, Anna Staudenmeyer, the deer biologist, and Wokin S. Fujihira, the biotech, are employed under the Department of Lands and Natural Resources. We are funded in part by the Department of Interior's U.S. Fish and Wildlife Service and the Department of Interior's Office of Insular Affairs. You may be wondering why we have a deer biologist, but I'll let Anna talk about that in the other part of the video. Okay, before we head out, a quick safety briefing for everybody and an overview of the plants that we work with. Many of them are found in the limestone forests or limestone flats. That's where we'll be heading. And the limestone here in Rhoda is exceptionally sharp and in many places brittle, so please watch your step. That being said, the karst of Rhoda will eat your boots for breakfast and spit out a shredded mess, so maybe bring 2 pairs. And if this doesn't deter you, Rhoda offers you the sasata or boonie bees, aka jungle paper wasps, which will not hesitate to sting you with their jungle concoctions if you come too close to their inconspicuous nests hanging under branches, so please try not to brush up against any trees. Let's move on to the plants. Here you can see the flowers and seed pods of Cereanthes nelsoni, or in the native Chamorro language, Tronconhuafi, or fire tree. This is the largest of the 3 plants we work with, and it's found at higher elevations around the island. This species is only found on the island of Rhoda and an island south of here called Guam, but unfortunately Guam has only 1 remaining naturally occurring tree, while Rhoda has over 50. A gauge, an idea of the size. Here we have some drone footage, and you can see the one of our work trucks popping in and out of the frame. So it kind of gives you an idea of the size. On this day, we were collecting canopy measurements, DBH, and seeds for propagation in the nursery. Next up, you can see the flowers and fruits of Osmoxylon marianense. This is endemic to Rhoda. It's found in the moist limestone forest areas, and it predominantly exists on the upper plateau of Sabana and its slopes. Here you can see a photo of one of the outplantings from the 90s, late 90s, that DLNR Rhoda Forestry outplanted. And this is a milestone in their program because it's producing viable seeds for our program now, which is pretty amazing. And last but not least, well, it is kind of the least in size, is Nassogenus rhotensis, which is another endemic Rhoda species, which is found on limestone flats. And here we have a video showing you where they're found. They're a little bit above sea level, several meters above sea level. And we've actually successfully propagated this plant several times from seed and are currently working to find suitable habitats to test outplanting feasibility. If that doesn't work out, we're considering doing direct seeding in suitable habitats. So this is just to give you an idea of some of the places we go and how far out they are away from the road and how rough the terrain is. And the sun is pretty intense here in the tropics, so sitting out here collecting seeds or measuring plants is kind of tough business. But let's enjoy our field trip today. Here we go. So while we're driving out to our location, I'll give you a little history of our program and how it got started. So anyways, in the late 80s, to address concerns of endangered plant species on Rhoda, the forestry section began work propagating, outplanting, and maintaining endangered species while monitoring the naturally occurring trees in the wild populations. Fast forward to 2015, with access to federal funds to initiate private landowner participation through a United States Fish and Wildlife Service Partners for Wildlife grant, the Department of Lands and Natural Resources Forestry section was able to onboard 2 volunteers and focus on increasing the number of individuals on private lands of these endangered species. And this ensures their protection and the buy-in of local stakeholders, in particular the community. In 2016, another grant was applied for with the U.S. Fish and Wildlife Service to bring on a full-time nursery technician to conduct these same activities with 3 specific plants in mind. And that's when I started in 2017 to continue the work of the previous volunteers. A majority of our outplantings are conducted on government lands, pretty deep in the jungle. So there are some access roads, but as you can see in this footage, the road is not that wide and it's not that smooth. These access roads get us to our outplanting, well, they get us to our parking lots, and then we must hike into our outplanting sites. To kind of give you an idea of this, we have some footage of that too. That being said, one of the biggest problems we have here on Rhoda with outplantings is plant pests, and one of them you'll see on your screen fairly shortly. A Philippine brown deer run across the screen. You may have just missed it. Look closely, here comes the fawn following the mother. These guys browse on our plants. The bucks rub their antlers on the plants, causing problems. They were introduced to the island of Guam and other islands in the Marianas by the Spaniards in the late 1700s. So we've arrived at our outplanting site, and we're tossing on our planting bags, and we're going to grab our tools and fencing supplies to install a few fences and 4 plants. We'll spare you the extra 3 plants, and we'll show you one of the Cereanthes. This is right off the road, actually. We drove the truck in here, but we had to hike in a little ways to get to the actual outplanting site, so we'll carry this equipment. If it was a larger outplanting, we would probably just take all the fencing first, install the 10 or 20 individual fencing locations, and then bring the plants in. Since we're only planning a total of 4, we could carry everything in. After we get done here, I'll stop talking. You can enjoy the sounds of rain, but we'll be taking you out to a grassland conversion site after here. Here we are at one of our other project sites. We're going to be hiking down to an area called Guadzaongan, and this is part of the Chonconguafi's historical range. The karst here is pretty bad, so we kind of take it slow, and then when we get to the open field, it's basically a big bald spot in the surrounding canopy. I'll be applying some slow release fertilizer pellets at each outplanting and doing some routine maintenance around the trees. And you'll see here in a few seconds the some of the outplantings here, and some of the fencing you'll notice is kind of off kilter, and that's from deer like running into them or something. I don't know for certain. Wilkin and Anna will be splitting off to retrieve one of their many cameras, and which brings us to why we have a deer biologist in our program. So I'll hand over the mic to her, and she's going to explain her aspect of the project on the deer side of the house.
Since James and Kendall have to spend so much time protecting the Cereanthes, Osmoxylon, etc. from the largest wild herbivore on Rhoda, deer, they brought me on to study them. This species of deer goes by many names. Its taxonomic name, Rusa Mariana, also known commonly as Philippine brown deer, and locally as the sandbar deer, or Banado in Chamorro. The main aim of my research is to estimate the abundance of deer on Rhoda so we can make informed adjustments to the deer management here that benefit the hunters, farmers, and the native species and ecosystems unique to the island that are being pressured by the overpopulation of a non-native herbivore. I'll be using data collected from our randomly placed camera sites across the island, as you can see Kendall and I working at in the video. This segment is showing just a snapshot of the camera setups that we use and the kinds of data we collect in the field to inform our models. Believe it or not, using distance reference videos like the one we are creating here, I can use images of the deer and how far they are from the camera to estimate how many deer there are on the island. Since their introduction to the island 200 plus years ago, little work had been done in the scientific community to study or manage the deer on Rhoda. But in that time, the species has become as ecologically detrimental as it is culturally significant, and is now a favorite game species on the island, which makes eradication an unfavorable option. So, my efforts within the Luda Plant Extinction Prevention Program aim to rectify the situation by both protecting the native and endangered plant species and allowing a huntable population of deer to persist on Rhoda. By understanding how many deer there are, we can adjust the current hunting regulations with the objective of reducing overall population numbers to a sustainable level. Ideally, if the overpopulation is brought into balance, then pressure on both native vegetation and agricultural crops should be alleviated to tolerable levels. With our cameras, we also collect a lot of interesting peeks into the life and life histories of Banado. Sometimes the deer find our cameras and check them out, and sometimes they just stop to give themselves a bath. To date, our cameras have captured many candid moments and important information about the life histories of these little studied deer, like a fawn nursing from its mother. Though these deer are non-native, an important step in managing any ungulate species requires understanding traits of their life histories, like how often they reproduce and how many offspring they have at a time, when the rut or the mating season occurs, and if it is seasonal or happens year-round. We can infer when the rut is occurring from videos of things like sparring, which you can see a clip of here, and you can also hear the very interesting whiny sounds that bucks make while they're sparring, and also in clips of bucks pursuing does that are, I'm assuming, in estrus. And by the stage of regrowth of the buck's antlers, we can also tell where they are in their mating cycle. This buck, you can see that his antlers are in the process of shedding. One of them is already dropped, and the other one will likely drop soon. To velveted antlers, just beginning the process of regrowing, and velveted antlers only a few weeks from beginning the process of shedding the velvet and beginning the rut. As you can see, both these bucks are at very different stages of antler regrowth, which we saw across camera sites and suggests asynchronous rutting of Philippine brown deer on Rhoda, which is a really interesting finding that we're getting from our camera data already. So, as the study proceeds, we hypothesize that we will see rutting behaviors, fawns, and all stages of antler growth occurring year-round. We're going to continue to collect information about the life histories of deer on Rhoda from video evidence, and gathering first-hand information from hunters and others out and about in the jungle to better understand and better manage the species. Our cameras also capture interesting feeding behaviors, like this buck working hard to consume a Barringtonia or puting seed, or standing on their hind legs to reach higher branches. We captured the eerie sound of a deer barking that anyone who has spent time in the jungle on Rhoda knows well, from both males and females. And when we got really lucky, the deer were nice enough to show us how they go to the bathroom, which unsurprisingly happens the same way as it does with any species of deer. At multiple sites, we also spotted deer and another introduced species, jungle fowl, becoming fast friends. And, at a handful of cameras, we were lucky enough to catch one of the rarest species of bird on island, the Aga, or Mariana crow. Our program works closely with the Aga Project on island. We share information, data about our bird sightings and band IDs. They tell us when they see some of our plants of interest or if they find a new naturally occurring plant, which is always exciting. And now we'll hand it off to the Aga crew so you can learn more about their project and what they do.
Approximately 2.5 million years ago, a crow that would one day be known to humans as the Aga, or Mariana crow, diverged from its most recent common ancestor. Over time, the Aga and its close relative, the large-billed crow, evolved marked differences in their physical characteristics and their geographic ranges. Where the large-billed crow is over twice the Aga's size and inhabits an enormous range spanning Central, Eastern, and Southeast Asia, the Aga is slight in stature and restricted solely to the Mariana Archipelago. The Aga is endemic to the islands of Rota and Guam, where native limestone forest is its preferred habitat. Although there is evidence that many of the forest birds in the Marianas once existed widely throughout the archipelago, there is no evidence that the Aga ever flew north of Rota. In the early 2000s, Guam's Aga population was extirpated by the introduced brown tree snake, leaving Rota as the only place in the world where Aga exist. There are no snakes on Rota, and yet still, over time, the Rota population has declined from more than 1,000 birds in 1982 to only 200 individuals at present. Since 2005, the University of Washington has been contracted by CNMI DLNR to monitor the Aga population and determine causes of decline. Our work has revealed that young Aga have high mortality rates, and that the major cause of mortality in juvenile crows is a multi-organ inflammatory response that leads to organ failure. What is causing this inflammation that leads to the death of so many Aga? It isn't clear yet. Viruses and bacteria have been ruled out, and the pathology does not appear to be infectious. Could it be a response to a foreign organism that is extremely difficult to detect? A toxin? Or perhaps a genetic disease? While the factors driving juvenile mortality will take more time to unravel, we have established an ongoing captive rear and release program towards the goal of reversing the Aga's declining population trend. Population models made in 2014 showed that increasing adult reproductive output via captive rear end release would be an effective way to supplement the wild Aga population on Rota. In 2017, San Diego Zoo Wildlife Alliance joined the University of Washington as partners in an ongoing population augmentation effort. The San Diego Zoo Wildlife Alliance crew incubates eggs taken from wild crows, rears juveniles to independence, then releases them into the wild. The University of Washington crew finds the Aga nests that have eggs or young nestlings, then monitors the captive reared birds post-release. This program works by effectively doubling the productivity of Aga pairs from which eggs are collected. Aga can nest nearly year-round and usually re-nest multiple times after failure. By collecting the first clutches of a breeding season to rear in captivity, the wild pair is then free to rear a second set of offspring on their own. Our program raises the captive Aga in large aviaries here on Rota until they are 1 to 2 years old. old enough to breed, and also old enough to be past the stage of high juvenile mortality associated with the poorly understood inflammatory response. Rearing young aga begins with incubating eggs, then progresses as the egg is moved into a temperature-controlled hatcher when the chick begins breaking out of its shell. Chicks work hard during the hatching process. As they work to get out of the egg, the blood vessels attaching them to the egg membrane diminish, and the young birds become dependent on parental care. Our program has been highly successful in hatching eggs and rearing nestlings to independence. Out of 51 viable eggs and nestlings that our program has taken in, 94% have been successfully reared to independence. Once their eyes open, chicks are fed with puppets behind one-way mirrors. This prevents young aga from imprinting on humans and also gives captive nestlings exposure to visual imagery of their own species. Avoiding imprinting is critical to their success after their release into the jungle, where they will live with wild crows and avoid human activity. The birds are given natural foraging opportunities and wild food items from a young age and are able to begin foraging immediately after release. To ease their transition into the wild, we do soft releases, meaning that we hold our birds in aviaries at the release site and then provide supplemental food for several weeks. All of the birds we've released have now been independent of supplemental food for a year or more, and many are paired and nesting in the wild. While wild juveniles have a 69% first-year survival rate, 87% of the birds reared by our program survived their first year post-release. Not only are the aga surviving, many are also nesting. Most of our released aga have paired with mates. 4 different pairs have built nests, and 2 captive reared aga are paired with wild mates. Here is one pair collecting coconut fibers they will use to line their nest. This released aga laid 2 eggs and faithfully incubated them with her mate. Sadly, the eggs never hatched. To date, no captive reared aga have successfully fledged young in the wild, but we are optimistic this milestone will soon occur. The worldwide population of this critically endangered species consists of 54 nesting pairs on the island of Rota. Nearly 10% of these birds are made up of aga reared by the San Diego Zoo Wildlife Alliance and released by the University of Washington. Right now, we currently have 26 additional captive reared birds in our aviaries, ready to be released in 2021 and 2022. Meanwhile, we are collaborating with experts across the world to determine the cause of the mysterious inflammation associated with mortality in juvenile aga, and we are working with Rota's community to increase local involvement and local ownership of the conservation of this incredibly unique species. The aga has been a part of the ecological community of the Marianas for more than 2 million years. We hope that with our work, we are helping the CNMI community retain and strengthen this longtime member of its natural world.
My name is Bradley Eichelberger, and I am the Chief Geographic Information Officer for the CNMI Division of Fish and Wildlife, and I'm here to talk today about our island-wide survey and translocation projects. Since the 1990s, the CNMI Division of Fish and Wildlife has been conducting biological surveys on all of its islands to monitor bird, fruit bat, reptile, land crab, and invasive species populations. These expeditions often entail extensive efforts to collect as much biological data as possible in a time frame of 2 to 3 weeks. Since there are 14 islands in the CNMI, our goal is to resurvey islands within a 10-year time frame in order to provide best wildlife management recommendations and practices. The information from such surveys are vital to monitor and assess impacts of invasive species, habitat loss, and climate change on our island's wildlife populations over time. One of our most recent expeditions was a 9-day, 10-person trip to the uninhabited volcanic island of Anatahan in May of 2021. Anatahan's volcano erupted in 2003, and there hasn't been an extensive set of biological surveys in over 20 years. When the island erupted, most of the vegetation was covered in ash, and the fate of the wildlife populations has largely been unknown. 18 years following the eruption, the island has had a chance to regenerate, and it remains a remarkable wonder to see with one's own eyes. The island itself is fairly impenetrable with steep ravines and high ridges, but we were able to collect a lot of data, including new species records for birds, insects, and plants that have never been documented there before. Here we see DFW biologist Emily Kohler conducting a 5-minute auditory survey for bird species at one of our survey stations. DFW biologist Bronson Curry is pictured conducting a survey that documents the presence or absence of the New Guinea flatworm, a parasitic flatworm that causes native snail mortality. DFW technician Keith Torres is seen here checking reptile traps along a rocky strand transect to document the presence and absence of lizards and assist the other biologists in collecting genetic samples for species identification as needed. We also use a series of vegetation plots to measure species cover, composition, and structure across the various plant community types. In addition to on-the-ground surveys, we were able to use unmanned aerial vehicles and geographic information systems to view the more remote areas of the island, such as this video of the crater of Anatahan, where we can actually see some vegetation regrowth. Not only do these surveys provide us with information about the wildlife populations and habitat trends over time, it also provides us with information for assessing the potential translocation of native species throughout the Mariana Islands. With the potential threat of invasion of the brown tree snake, an invasive exotic species that has decimated bird populations on the neighboring island of Guam, the CNMI DFW partnered with the United States Fish and Wildlife Service, Pacific Bird Conservation, and the Association of Zoos and Aquariums to create the Mariana Avifauna Conservation Project, or also known as the MAC Project. The primary goal of the MAC Project is to translocate native birds from the more invasion-susceptible southern islands in the CNMI to the more isolated northern islands. Almost all of the proposed species for translocation are endemic to the Mariana Islands, meaning that they are not found anywhere else in the entire world, and some of these species are only found on very specific islands within the island chain. For example, the Tinian monarch, found only on Tinian, was translocated to Guguan in 2017, and the golden white-eye, found only on Saipan and Agigan, was translocated to Sarigan in 2012 and Alamagan in 2018. These translocations and the resulting satellite populations are an essential tool to prevent the extinction of these endemic species should the brown tree snake begin to colonize the lower southern islands. As mentioned before, once an island-wide survey has been conducted, DFW biologists decide which species are the most appropriate to translocate to the islands of interest based on determining suitable habitat from the vegetation surveys and using the results from the bird surveys to determine if there will be competition among the translocated species and the birds already occurring on the island. Translocated birds are typically misnetted on either Saipan or Tinian, fed a diet to ensure their health is suitable for translocation, tested for parasites by trained veterinarians, banded with a unique color combination to ensure individuals can be identified by sight, and released to the appropriate island the year following an island-wide survey. DFW biologists then resurvey islands following translocations to determine the translocated species' survival and reproduction success. Since all of the translocated birds were banded prior to release, DFW biologists will try to re-site birds and identify them by the specific color combination of the band in order to assess survival. Additionally, any unbanded birds within the translocated species that are encountered are recorded, since this indicates that the species has reproduced and serves as a good indicator of reproduction and translocation success. This concludes my portion of the virtual tour, and the CNMI DFW appreciates your interest in our conservation efforts and research projects. For more information, please check out our website at www.dfwcnmi.com or check out our Facebook page at facebook.com/CNMI-DFW. For more information regarding Pacific bird conservation and the Mariana Avifauna Conservation Project, please visit www.pacificbirdconservation.org. Thank you again.
Join us on a trip with our sea turtle crew. Green sea turtles are found in tropical and subtropical waters worldwide and have been of conservation concern for decades. Their life history traits such as being long-lived, maturing later in life, being highly migratory, all make them vulnerable to anthropogenic impacts on land and at sea. The recent status review under the U.S. Endangered Species Act suggested that 11 distinct population segments exist for green sea turtles worldwide. While several regions, such as the North Atlantic, which encompasses Florida, Gulf of Mexico, and the Caribbean, and the Central North Pacific, covering Hawaii, have populations with recovering trajectories that are listed as threatened. The population in the Central West Pacific, which includes the Northern Mariana Islands, are listed as endangered due to population declines or are data deficient. The regional population of green sea turtles in the Marianas was listed as endangered in 2015. The major threats to the green sea turtles in the CNMI are illegal hunting and climate change. Ongoing monitoring indicates that the small population of endangered green sea turtles in the Western Pacific has seen at least 1/3 of its nesting females removed from their nesting beaches in the past decade. And now, with nest temperatures increasing, turtles are being pushed toward temperature tipping points. In sea turtles, the temperature of the nest determines the embryo's sex, so warmer temperatures are producing more females, and at some point, hatchling survival will decrease as well. To tackle these issues, the CNMI's DLNR, in partnership with NOAA and other scientists, have been tagging and monitoring nests and conducting outreach and education to use the cultural significance of sea turtles to shift the traditional consumption of turtles to a conservation narrative. Reports from the public play a key role in our response to both sea turtle presence and illegal harvest or harassment. It's important to note that it is illegal to harvest or harass sea turtles, and all activities shown here were conducted under permits by the U.S. Fish and Wildlife Service. We have developed a K-6 curriculum that integrates with school standards. We also reach out to the public through media and educational events. Green sea turtles come to our beaches to nest, where we tag the nesting females, monitor the temperature of the nests, and assess nest success. Green sea turtles lay about 80 to 120 eggs per nest, which takes about 55 to 70 days to hatch. Once the eggs hatch, hatchlings pool and escape to the ocean. Here, the public reported and recorded a daytime hatching. 48 to 72 hours after hatch, we conduct a nest inventory. During these nest inventories, eggshells are counted and development of unhatched eggs is determined. Our team's presence on the beaches not only allows use to collect data, but our presence deters poachers. However, poaching still occurs, and we respond to reports on our hotline. An example of this hotline being effective in preventing sea turtle poaching was when our partners at BECQ reported a nesting female that was dragged more than a mile from the parking lot. As you can see by these marks, she was dragged quite a ways, not only along the beach, but on the road as well. With help from these volunteers, we managed to rescue her and get her back in the water. This sea turtle is nicknamed the Warrior. She survived not one, but 2 poaching attempts. While our efforts to stop poaching on the beach continue, education and outreach are also critical to saving our sea turtles. Overall, our research monitoring and outreach efforts help resource managers work to protect turtles and their habitats. Ultimately, the goal is to support species recovery.
As a response to 2 super typhoons that passed through the Mariana Islands, the Mariana Islands Nature Alliance proposed a tree planting campaign, Bring Back Our Trees, to revegetate the coastlines of Saipan to prevent further sedimentation and runoff into the lagoon, leading to degradation of our coral reefs. Bring Back Our Trees proposed to plant 300 native trees along the west side of Saipan, but completed the project planting over 700 trees throughout the coast of Saipan and at one site on Tinian. In 2015 and 2018, Super Typhoon Sulor and Yutu passed through the Marianas, creating havoc towards our ecosystem, specifically destruction to our trees, native and introduced. The loss of trees jeopardized our island's environmental health by leaving our shores vulnerable to erosion and sedimentation. MINA implemented a tree revegetation campaign to mitigate the damages of the super typhoons to support a more resilient, natural environment and help prevent sedimentation and further degradation of our reefs. The tree planting was done throughout Saipan's public parks and coasts. By NOAA's definition, the only difference between a hurricane and a typhoon is the location where the storm occurs. A typhoon impacts the environment through the damages to vegetation, agriculture, flooding, which eventually leads to soil erosion, higher coastal tides, storm surges, and sedimentation.
Prior to the recent disasters and the current pandemic, the eastern coastline of Saipan experienced an increased number of visitors, particularly at Jeffreys Beach. Once tourism opens up in the CNMI, it is anticipated that this trend will resume. The CNMI government is also working to complete a coastal highway project on Route 36, which will connect this rural setting to the scenic areas in northern Saipan. Jeffreys Beach is located at the mouth of the Talakhaya streambed and has undergone heavy visitor use despite difficulty with access. The roadway to Jeffreys Beach is heavily degraded and is unsafe for visitors and local residents due to the physical conditions. This dirt road runs parallel to a streambed that provides habitat for endangered fauna. The shoreline is only accessible by foot or by vehicle with high clearance. Restoration of the roadway would reduce sediment runoff into the streambed and protect sensitive habitat.
The Bureau of Environmental and Coastal Quality's Division of Coastal Resources Management acknowledged the need to enhance public access in the area in an effort to provide a unique attraction that would serve as a tourist destination, enrich the experience of its visitors, and preserve sensitive geographical and cultural features in the area. DCRM capitalized on this opportunity and worked with a local company and 15 architects to develop an ADA-compliant walkway that will stretch from the main road to the shoreline. The boardwalk concept has had reasonable success in Saipan, as evidenced by the Beach Road pathway and the Calavera Cave redevelopment. This boardwalk shall serve as a pilot project for future public enhancement improvements that, if successful, shall be replicated for other beaches on the eastern coastline of Saipan. The design for Jeffreys Beach shall incorporate amenities that consider the natural environment, such as permeable parking, 2 viewing platforms, educational signage, trash receptacles, and a restroom facility. It is also DCRM's intention to utilize construction material that is durable enough to withstand a harsh tropical environment.
The CNMI is working hard to conserve and restore our terrestrial as well as our ocean resources. While our landmass is comparatively small, we are a big ocean state with an exclusive economic zone of over 1 million square nautical miles. The indigenous people of the CNMI have a deep and cultural connection with the ocean, as it is a source of traditional knowledge and practices passed down through the generations. Additionally, the CNMI's ocean economy employs up to 8,300 people annually, which is roughly 40 to 50% of our local employment, contributing about $922 million annually towards the CNMI's gross domestic product. Despite the significant economic and non-economic values of our ocean resources and local commitments to managing and conserving these resources, these systems are suffering from impacts from global climate change, as well as local stressors that present management challenges. Saipan has the most diverse types of coral reefs and associated habitats in the CNMI. A fringing and barrier reef system protects the majority of the beaches along the western side of the island, forming a 12.4 square mile shallow water lagoon system. This environment contains large assemblages of seagrasses, coral reef habitat, and the last remaining mangroves in the CNMI. The western side of the island is the most populated, and the coral reefs along these areas are often negatively affected by human activity. Continuing sediment and nutrient pollution, combined with sporadic stressors such as outbreaks of crown-of-thorns sea stars or COTS, thermal stress and bleaching, and more, affect many of western Saipan and southeastern reefs. To combat these stressors and increasing threats from ocean acidification and rising temperatures, several complementary coral restoration efforts are underway with a focus on the Saipan Lagoon. Dr. Eliza Johnston, who is leading innovative restoration efforts in CNMI, will share more about these challenges and solutions being implemented with us on our virtual tour today.
Okay, so unfortunately, as many of you know, coral reefs are in crisis worldwide. Coral reefs are declining in every region that they exist, and this is due to a variety of local and global scale stressors. These include coastal development and land-based pollution, including nutrients and other chemical pollutants, plastics and marine debris, sedimentation and habitat loss, acute disturbances such as ship groundings, predator outbreaks, and severe weather events, overharvesting of marine organisms, especially herbivorous fishes, which are really important to reef health. And then other impacts from human use, like sunscreens, turbidity, physical damage from anchors and people standing and kicking corals, where human activity is high. And then, while all of these local stressors are really important and are negatively impacting coral reef habitats everywhere where humans exist in close proximity to reefs, including the CNMI. The biggest, the largest threat to coral reefs currently is global climate change, and specifically ocean warming. And this occurs because when the water gets too warm, corals will get stressed out and they bleach, and if the water continues to stay warm, they will die. And so what is coral bleaching? It is not, someone out there with Clorox, pouring Clorox onto the reef. Corals have a symbiotic relationship with these microscopic algae called Symbiodinium or zooxanthellae. And so the zooxanthellae live within the tissues of the coral. You can see here. And normally the algae can provide up to over 90% of the energy that a coral needs to survive and grow. And they produce these products through photosynthesis. And the zooxanthellae also give the corals their color. So when the water gets too warm, this symbiotic relationship breaks down, and essentially the symbionts are expelled from the coral. And what you're seeing is it appears very bright white, and what you're seeing is the skeleton underneath. When they first bleach, the corals are still alive. There's still a thin layer of tissue on the skeleton, but it's clear, much like their cousins, the jellyfish, which we're used to seeing. The tissue of a coral animal is clear, and so you see the bright white skeleton underneath. Corals can recover at this stage if the stressor is reduced. So if the water temperature cools down within a pretty short amount of time, they can recover. However, if it stays warm, they will die, and then the skeleton gets overgrown by turf algae, which you see in the sort of left panel of this infographic. And so that's been happening all over the world. We've had some major global scale bleaching events, and these events are increasing in scale and severity with climate change as we put carbon dioxide and other greenhouse gases into the atmosphere, and the planet gets warmer. The oceans have actually absorbed over 90% of the heat created by humans since the 1970s. So the surface seawater temperatures are increasing as well, causing these frequent and severe bleaching events. So the last one began, here it says 2015, but we actually started seeing the effects in the CNMI in 2013. So from 2013 to 2017, we had 4 near consecutive thermal stress and bleaching events, 3 of which were very severe, which you can see here in 2013, 2014, and 2017. In 2013 and 2014, the Saipan Lagoon was impacted pretty severely, as well as a lot of the northern islands, like Mog, Pagan, Sarigan. The coral reefs up there in 2013 and 2014 got hit pretty hard. And then in 2017 was actually when the Saipan experienced the most severe bleaching event that we've seen here on record. And this resulted over 60% loss of shallow water corals. Just to demonstrate this, we surveyed in 2018. We surveyed 34 reef sites all around Saipan that we had previously surveyed in 2012, and you can see most of the sites experienced over 40% relative loss in coral cover. Many of the sites, these red sites, experienced over 80% loss of corals. And some taxa, some species, like the Acropora species and Poecilopora species, these are the sort of branching guys that you see a lot on the shallow reefs. Little digit-shaped cauliflower-looking corals. They were hit particularly hard. Some sites we lost over 90% of Acropora species and 75% of Poecilopora species. And so that led to, sort of drastic changes in the coral reef structure and community composition around Saipan. And so, as we all know, living in these islands, coral reefs are very important to island communities. They're important for food. The communities here rely on reefs for sustenance through fishing. Also for income, they rely on them for income through, tourism activities, and coastal protection. So reefs are very important for dissipating wave energy during storm events. So all of these tropical storms and typhoons that come through, these reefs really protect the coastal environment from even worse floods and wave damage. We also use the reefs for, cultural and recreational activities. And so they're very important. And the local management agencies, seeing all this mortality over a very short period of time, realized that we really needed to start taking some active measures to mitigate these losses. And while threat reduction remains to be really important on both the local and global scale, we need to reduce these stressors so that these reefs can remain resilient in the face of ongoing global climate change. Active habitat restoration has also emerged as a really important tool for local managers and conservation organizations to try to, keep these habitats alive and mitigate losses of ecosystem services. And I just wanted to point out that it's very important to, when we're thinking about habitat restoration and developing our restoration program, that we emphasize improving the resilience of these reefs to withstand the global climate change that is occurring and will likely continue to occur throughout the near future. So the rationale between restoration, again, is to jumpstart the recovery of these really important ecosystems, mitigate the loss of ecosystem services, improve resilience of corals and ecosystems, maintain important biological and ecological processes, create opportunities for local community engagement, provide research opportunities, and create and maintain a coral gene bank. And so there are many approaches to coral reef restoration. Most of them these days involve some type of coral propagation. So propagation is just essentially creating many new organisms from a smaller number of parent or donor colonies. And corals, much like plants, can be propagated asexually and sexually. They're essentially clonal organisms made up of tiny little polyps that when you cut a little fragment off, you can see this colony on the right. There's a little baby fragment on the right, and the bigger sort of donor colony in the nursery on the left that we had just fragmented. And so you can cut off these little pieces, and much like many plants, they will continue to grow into a new coral. And so this is a really popular tool in restoration programs. It's often referred to as coral gardening, and it's most of the time these coral fragments are propagated within nurseries where they're in a sort of a protected environment where they can be, maintained and cared for. You weed them essentially, you clean all the algae off their trees, and you take good care of them until they get to a size where they're more likely to survive out in the wild, and then you outplant them onto these degraded reefs. This is valuable in that you can create many corals from a few, again, minimizing the impacts to wild donor populations. However, you're inherently sort of limited in scale in that there's only so much space you can put nurseries. It's very time intensive to go out there and fragment these corals and take care of them and then outplant them onto reefs, where you essentially have to paste every individual coral down onto the reef. And so you're limited in spatial scale, and if the scale coral reefs are dying, that's going to be an issue in the future. You're also limited in your genetic diversity. So each one of these new little fragments is a genetic clone of the parent colony that you cut it from. And so you can only house so many genotypes, even though we try to maximize the diversity within our nurseries. Again, you're limited by space and resources. So your genetic diversity is inherently limited as well, and that in turn limits the ability of these corals to adapt to a changing environment. So another important way to propagate corals is through sexual propagation. But essentially you're collecting the gametes. Many corals will spawn eggs and sperm, and you can collect them. You can assist fertilization, and you culture the larvae through to settlement. You then can maintain, these juvenile corals, again, in a nursery setting or some sort of controlled environment for a little while, or you can directly seed them onto the reef. And so this method it reduces mortality of early life stages, and you can really harness the huge amount of reproductive output that corals produce. And so it has the potential to greatly increase restoration scale and also genetic diversity and associated adaptive capacity. So each little baby coral shown here on the right that is produced through sexual propagation is genetically unique. So you can end up, with thousands or even millions of genetically unique new colonies, which is going to be really important for the health of these ecosystems in the face of ongoing climate change. And so, however, there are limits to this method as well. Gamete collection and larval culture can be logistically challenging, and outplanting is still inefficient, and long-term survival is often low. So the CNMI has already gotten a start on asexual propagation methods. In 2019, the Saipan Coral Nursery Pilot Project was established. This was funded by NOAA, Pyro Division, contracted to us at JAMS, and it was really a collaboration between federal, local agencies, and the private sector. So it was a really cool project to work on and continue to work on. And we really try to collect a lot of data about these corals and the environment, both at the nursery and at our outplanting sites, so that we can, learn about how to make these restoration efforts successful and adapt our strategies as we go. So currently, we have 20 tree structures out at the nursery with about 1,200 fragments from 9 species and 4 genera. We currently use 2 fragmentation or asexual propagation techniques, and those include microfragmentation, shown in this panel at the bottom, and then are just the sort of traditional fragmentation techniques of just cutting off pieces of the coral and making new corals. Also, DCRM, the local Coastal Resources Management Office, recently installed, I think in the last few months, the second coral nursery in the Saipan Lagoon. And this nursery has capacity to house over 1,000 coral fragments. And both of these nurseries will continue to grow and diversify over the coming years. So that's really exciting. And we did a pilot outplanting last year, and we'll be doing 3 more outplantings this year, hopefully. So it'll be really cool to see how these guys do out on these reefs. But we know, because of the limitations of asexual fragmentation, we knew we really wanted to start incorporating sexual propagation into our restoration program. So last year, when NOAA put out a request for proposals for the Ruth Gates Coral Restoration Innovation Grants, this funding was to enhance coral resilience and improve the long-term success and efficiency of shallow water coral restoration in a changing climate. So I thought this would be a good opportunity to sort of get some money and build capacity in the CNMI to start incorporating sexual propagation techniques and also start, attacking some of these challenges that still exist with coral restoration. And these include, as I mentioned, scale and long-term survival and success of outplants, which resilience of these corals and ecosystems is a really important factor in those issues. So I started thinking, I was very familiar already with the wonderful work of CCOR, but CCOR for a long time has been working on developing these settlement substrates over here on the left called, they're tetrapods. The first version, there are some different shapes now, but they are designed to self-stabilize in the reef without adhesives. So you settle coral larvae, just briefly, you settle coral larvae on these substrates, and you can quickly put them out onto the reef by wedging them into little nooks and crannies in the reef substrate. And this picture on the right is pretty exciting because this is actually a picture from Guam, from a baby that was outplanted on one of these substrates in 2017 through the Guam Coral Restoration Initiatives over there. So this process is pretty effective. Currently, I think they just do this hand placement method, but the idea was to be able to upscale this and disperse a lot of these over a reef, like from the back of a boat or even using drones or something like that. But we haven't gotten there quite yet, and there are similar approaches. I was thinking about similar approaches also on land. So on land, seed balms, or essentially these balls of soil and seeds, have been used for forest restoration for a while as well, at various spatial scales and using various levels of technology. So I was thinking about how we could build on, these methods, what CCOR has been doing already, and I decided that maybe we needed to think smaller, much smaller. So I developed these very early prototypes of what became what we started calling larval seed beads. And they're essentially just, these are colored, by the way, these were colored just for easier visual detection, to demonstrate both the size of these substrates that I had in mind and their ability, again, to self-stabilize in these really abundant little nooks and crannies and niches that are all over the reef framework. And so reefs have these little holes and grooves, like very small ones. Even reefs that are relatively low in structural complexity will have these little holes in them. And so I thought if we could make smaller substrates, these would be easier to handle, easier to distribute over the reef in bulk, and that they would be pretty unobtrusive. So as you can see, these guys go into these little grooves nicely. They're very small. They're unlikely to do any harm to the natural environments. And if recruitment is not successful and you just have a bare bead, these things are small enough that they're going to be quickly overgrown by CCA and cemented into the reef framework without causing any harm to the ecosystem, even in large numbers. And so they're also small enough that we could easily distribute them across large scales of reef using things like, both aerial and underwater drones, boats, even small planes or helicopters to reach areas of reef that are hard to access via boat. So there's a lot of potential in just sort of shrinking these things down. I contacted HART to see if CCOR would be interested in collaborating on this grant proposal for the Ruth Gates funding opportunity. And luckily, they were interested, and so we wrote a proposal and submitted it, and ultimately we were selected as one of 4 recipients of this first round of Ruth Gates restoration grants. So that was very exciting, and the project, so we now have this funded project, and the title is Sowing the Seeds of Success, Testing Novel Approaches to improve the efficiency of coral reef restoration using sexually propagated coral. A 3-year project, and there are 3 main objectives. The first one is to develop and test novel settlement substrates designed to improve the efficiency of outplanting or seeding of sexually propagated juvenile corals at scale. Build local capacity in the CNMI to implement coral sexual propagation methods as part of a broader restoration strategy. And 3, test existing island-wide resilience assessment as a framework for increasing larval outplant survivorship. And so currently, early this year, we started with Objective 1. And this was a pretty interesting and exciting process to develop these little substrates. It mostly involved me drawing crazy pictures, bad sketches, and then Miles, creating these really cool 3D models and engineering schematics. And so we went back and forth a bit, and we narrowed it down to 5 designs that we wanted to put forth to potential manufacturers to get a quote. Based on those quotes, price points, feasibility, timeline, etc., we ended up narrowing it down to a manufacturer and 3 designs that we wanted to move forward with for this, for testing for this first year of spawning. And so we have these 3 designs that we lovingly call the Death Star, Mini Gear, and Gear Pod. And so the picture on the right is just the produced substrates with a current traditional Seacor settlement unit, just to show you the difference in size of these settlement units. And so we're actually going to test all of these in conjunction this year with spawning. So now we just need to add some larvae, and that brings us to the second objective of this project. And we are hoping that many of you will be able to come out and help us with our first year of spawning activities and get some hands-on training and learn how to do this so that we can keep using these methods and technologies in the CNMI at our local agencies and in our NGOs.
Together, all of these ongoing multi-stakeholder conservation efforts that we have highlighted today are supporting conservation of coastal resources on land and in our waters. Many of the implementation projects you have heard about today connect directly to long-term planning efforts being led by the Office of Planning and Development under the Office of the Governor to achieve critical objectives of the Global Sustainable Development Goals, including Goal 13, supporting urgent climate action, Goal 14, conserving and sustainably using the oceans, seas, and marine resources, and Goal 15, protecting, restoring, and promoting sustainable use of terrestrial and nearshore ecosystems, sustainably managing forests, and preserving and halting degradation of biodiversity and resources of particular concern in the CNMI. These natural resource-focused goals and supporting objectives further connect to goals for the built environment, such as Goal 6, ensuring clean potable water supplies, and Goal 11, promoting sustainable community development, as well as socioeconomic goals such as Goal 1, ending poverty, Goal 2, ending hunger, and Goal 3, ensuring healthy lives and promoting well-being for all at all ages. Leveraging partnerships, the focus of Goal 17 is critical to sustainable resource management and our shared mission to build resiliency of natural, built, and human systems for current and future generations. The Office of the Governor's Office of Planning and Development is charting a path ahead with a comprehensive sustainable development plan that supports sustainable use of marine and terrestrial resources. Please visit our website to learn more about planning and implementation efforts at opd.gov.mp. Thank you, Sidzuus Maasi and Olomwai, for taking the time to join us on this virtual tour. Your live Q&A session will now begin, where you're encouraged to ask questions.